Enzyme reversibility. Most enzyme reactions are reversible. The product synthesized by an enzyme can be decomposed by the same enzyme. An enzyme that synthesizes maltose from glucose can also hydrolyze maltose back to glucose. Enzyme action. Enzyme action and the reaction rate depend on several environmental factors, including temperature, pH, and the concentration of enzyme and substrate. Effects of temperature. In general, as the temperature increases, the rate of enzyme action increases until an optimum temperature is reached. Beyond optimal temperature, heat alters the shape of the active site of the enzyme and deactivates it, leading to a rapid drop rate. Effects of pH. For each enzyme, there is an optimal pH above and below which enzymatic activity declines. Maximal activity of many human enzymatic activity declines. Maximum activity of many human enzymes occur around pH 7.2, which is the pH of most body fluids. Exceptions include pepsin, which works best in the highly acidic conditions of the stomach, where pH is equal to 2, and the pancreatic enzymes, which work optimally in the alkaline conditions of the small intestine, pH of 8.5. In most cases, the optimal pH matches the conditions under which the enzyme operates. Effects of concentration. The concentration of substrate and enzyme greatly affects the reaction rate. When the concentration of both enzyme and substrate are low, many of the active sites on the enzyme are unoccupied, and the reaction rate is low. Increasing the substrate concentration will increase the reaction rate until all of the active sites are occupied. After this point, further increase in substrate concentration will not increase the reaction rate, and the reaction rate would have reached Vmax. Examples of enzyme activity. Every reaction in the body is regulated by enzymes. Some of the basic reaction types are listed here. 1. Hydrolysis. Hydrolysis reactions function to digest large molecules into smaller components. Lactose hydrolyzes lactose. Lactase hydrolyzes lactose to the monosaccharides glucose and galactose. Proteases degrade proteins into amino acids, and lipases break down lipids to fatty acids and glycerol. In multicellular organisms, digestion can begin outside of the cells in the gut. Other hydrolytic reactions occur within the cell. Two, synthesis. Synthesis reactions include dehydration and can be catalyzed by the same enzymes as hydrolysis reactions, but the direction of the reactions are reversed. These reactions occur in different parts of the cell. For example, protein synthesis occurs in the ribosomes and involves dehydration synthesis between amino acids. Synthesis is required for growth, repair, regulation, protection, and production of food reserves such as fat and glycogen by the cell. The survival of an organism depends on its ability to ingest substances that it needs but cannot synthesize. Once ingested, these substances are converted into useful products. Certain vitamin cofactors and essential amino acids cannot be synthesized by humans. If they are not available in a diet, deficiency and diseases as a result occur. Cofactors. Many enzymes require the incorporation of a non-protein molecule to become active. These molecules are called cofactors and can be metal, like zinc, iron, or smaller organic groups called coenzymes. Most coenzymes cannot be synthesized by the body and are obtained from the diet as vitamin derivatives. Cofactors that bind to the enzymes by strong covalent bonds are called prosthetic groups.